webinar in our series on animal-free recombinant antibodies. This series is being co-organized by the U.S. National Toxicology Program Interagency Center for the Evaluation of Alternative Toxicological Methods, or NICETM, the European Union Reference Laboratory for Alternatives to Animal Testing, and the PETA International Science Consortium. My name is Catherine Groff. I'm an advisor to the PETA International Science Consortium, and I'll be co-moderating today's webinar with David Allen, who is the president at Integrated Laboratory Systems and principal investigator of their contract supporting NICETM. We have two speakers today who will be presenting on the application of animal-free recombinant antibodies. First, we'll hear from Dr. Pierre Cosson of the University of Geneva, and then from Dr. Michael Fiebig of Absolute Antibody. Before I introduce our first speaker, I'd like to point out that a recording of this webinar will be posted at the link on the screen shortly after its completion. Information about additional webinars in the series can also be found at this link. There'll be time for questions for both speakers at the end of the second presentation. Everyone is on mute, but you can type questions or comments in the questions section within your GoToWebinar toolbar at any time during the presentations, and then we'll ask them at the end. This toolbar should be on the right-hand side of your screen. Speaking first today will be Dr. Pierre Cosson. Dr. Cosson is a professor in the Department of Cell Physiology and Metabolism and holds the Dorenkamp-Zimden Chair for Alternative Methods at the University of Geneva. At the university, he founded a recombinant antibody center focused on giving academics access to in vitro generated antibodies. Prior to the University of Geneva, Pierre performed research in cell biology at the European Molecular Biology Laboratory in Germany, the Centre d'Immunologie de Marseille-Luminy in France, the National Institutes of Health in the US, and the Basel Institute for Immunology in Switzerland. It's now my pleasure to turn the floor over to Pierre.
Thank you, Catherine. Can everybody hear me correctly? No shouts, so I'm assuming yes. So I must say that I'm very grateful for this occasion to talk and to present our work. I'm also a little bit intimidated because it's my first webinar, and I'm doing it from home where I'm confined with a COVID positive child. So these are peculiar situations, interesting times. The aim of this presentation is not so much to focus on theory of recombinant antibodies, but rather on the practical aspects about how they can be used, and particularly for people from academia. I will just start with a few words on the science crisis, and then most of my seminar will be devoted to how to solve it and how it can be solved with recombinant antibodies, and more specifically, how recombinant antibodies can help you do your work. And I'm going to focus on the very practical parts of all these questions. So let me start with the life science crisis. This is a paper published in 2012, where many of you have heard about it, where people at Amgen company try to replicate a number of high-impact original research studies that have been published in the best journals. They put all their efforts into it, and they really tried very hard. And in the end, 6 of the studies were confirmed, which means 11% reproducibility. And this is the life science crisis in one figure. If only 10% of our experiments can be reproduced, then the basis itself of science, reproducibility, is at risk. One of the main reasons for this crisis in reproducibility is that sometimes reagents are of extremely variable quality. And one of the worst reagents that you can find are antibodies. This was advocated in several papers in Nature in 2015, like the one that I'm here featuring, Blame It on the Antibodies, which made a long list of why antibodies are extremely poorly defined reagents with very little availability to all researchers and create enormous problems for reproducibility. Basically, if I do an experiment with an antibody and you want to reproduce my experiment, usually you won't have my antibodies available, and especially if it's in 10 years time from now, and that will make it extremely difficult for you to try to reproduce. One of the chilling figures that comes from a number of studies is that approximately 50% of commercial antibodies, so that's supposed to be high quality antibodies, do not recognize their intended target, or they lack specificity. It's probably worse with antibodies that are generated in individual laboratories, where they are stored in and distributed to small lists of people. Another paper the same year came to the conclusion that we absolutely must standardize research antibodies. This is one of the key elements if we want to improve reproducibility in life sciences. And the way to do that is to switch to recombinant antibodies. So I'm assuming that you all know what recombinant antibodies are, and you're already convinced that they are fantastic reagents. But let me just take one slide to summarize this. There are many reasons why you should switch to recombinant antibodies. There are defined reagents. A recombinant antibody is an antibody for which the sequence is known, so you know exactly what you're putting in your test tube. They're available in unlimited amounts. There are no limits to the amounts that can be produced, and they cannot be lost in time. You cannot lose a sequence that had been stored in databases. When you select antibodies, recombinant antibodies, you have a selection in controlled conditions, contrary to what happens when you inject an antigen to an animal, and then the black box of the immune system gets to work on it to select antibodies. And finally, of course, and this is a significant achievement in itself, recombinant antibodies can all be produced without the use of animals. Now, the key question is, how do you get access? And especially, how do you get access if you're working in academia and if you don't have unlimited funds to acquire antibodies? I'm going to describe the project that we have been developing in Geneva, ABCD project. ABCD stands for Antibodies Chemically Defined, which is really focused on providing access for academia to recombinant antibodies. Before I start getting into the details, let me just reiterate that this is an academia to academia project. So we are an academic entity. We're not for profit. Everything that we're doing is as open access as can be, which means almost purely open access. And everything that I'm describing is animal free. So the ABCD database is created to answer the first question, which is, well, I mean, you are maybe interested in using recombinant antibodies. You have a few favorite proteins. Are there antibodies that recognize your favorite proteins? In order to answer this question, we have created the ABCD database. It's hosted by the XPASI servers, so linked to UniProt. And there are currently 21,000 entries, 21,000 different antibodies that recognize a little bit over 3,000 different antigens. For each antibody, there is a unique identifier. It is linked to UniProt or to the library of chemical structures. There are detailed information, I will show you some, and you can order it. So the first question is, are there antibodies, recombinant antibodies already existing against your favorite database of your favorite protein? So if you want to get the answer, you log onto the ABCD database, and this is the address in the XPASI server in Geneva. If you click onto this link, you will reach this page. So as you can see, there is a lot of text, and there is a little box here that allows you to search by name or by keywords your favorite protein. So typically, let's focus on that. And let's say that you're among the people who today are interested in this brand new CoV-2 virus that we're all devoting so much time to. So you can access antibodies against CoV-2. Remember that CoV-2 was discovered less than a year ago. So getting antibodies available and tested today is already an achievement. If you type spike COV2 to get antibodies against the spike protein of COV2, or if you type the uniprot number of this protein, you will get a list of the antibodies, recombinant antibodies, that are available against the COV2 protein, the COV2S protein. You, by the way, see that each one of them comes with its unique identifier here, AI334, AQ806, etc. The list is actually very long. There are something like 120 antibodies against the spike protein, developed by very different groups around the world, and the information is all available. Yes, here 662 actually. It's in the database. Now, this is all nice and good. You know that your favorite protein, there is at least one antibody against it. So what information can you get about each of these antibodies? If you click on one of them, so this was the first of the list, AI334, this is the information that you can get in the database. So what is the antigen? It's a protein. These are the uniprot numbers. In that case, actually, and that's the reason why this was the first in the list, first in actually order of appearance, it was developed several years ago against a COV1 protein and turned out to cross-react with a COV2 protein. So you have two uniprot links in that case. This is the description of the antigen. This is the description of the epitope on the antigen. There are some details. And then you find here information about in what conditions it's supposed to work and where is the information coming from. Note that in that case, this came from a patent. So a lot of data in the ABCD database is published information, public information. Some of it comes from patents. A lot of it comes manual curation of the literature, which is an enormous amount of work. Every entry has been manually curated. And here, look, you have, or you're very lucky because you have clicked on an antibody for which there are reports in Antibody Reports. What is Antibody Reports? Antibody Reports is a journal that has been created by us. It's a fully open access journal and is devoted to the characterization of recombinant antibodies and only recombinant antibodies. And this is what an article looks like in Antibody Reports. You have here the title. These antibodies recognize the S protein, spike S protein from SARS-CoV-2 by immunofluorescence. And you can find a description of the antibody, how it's done, how it was the experiment done. And you see here the immunofluorescence pictures that show you that, yes, indeed, this antibody recognizes the S protein of SARS-CoV-2 in transfected cells. So not only you know that there is an antibody, that it works, but it has been actually retested in these exact conditions with this exact same antibody, and it works by immunofluorescence. Antibody Reports, I should spend a minute on it, is a journal which aims at describing how each antibody can be used. So there are lots of different articles, and the typical title is Antibody X recognizes protein Y in this technique, immunofluorescence, Western blot, ELISA, etc., etc. Lots of nice pictures around in these articles. And by the way, we also publish negative results. This antibody does not work in Western blot, or because this is almost as important to know as what are the conditions where it's known to work. So now that you know that you have all the information about this antibody, this is the antibody you want, how can we produce it? So you can order this antibody, but in order to be able to order it, you need to understand what format will be used for production. This is the structure of an IgG antibody, 2 heavy chains, 2 light chains, and here in orange, the regions that bind the antigen. This is the production format, which is adopted in Geneva, a standard mini-body format where you have the 2 variable domains, with the antigen binding site attached to the Fc portion, so it's a dimeric antibody like a real IgG. But think of it, you have the choice of the Fc portion. So it can be a rabbit Fc, a mouse Fc, a human Fc, guinea pig, etc., etc. The list is pretty long actually of all the possibilities. Typically, we have developed procedures for producing small amounts of antibody, 5 mL at approximately 50 micrograms per ml, so a total of 200, 250 micrograms in general. And you will get this production to you in 2 to 4 weeks. So the only thing you have to do basically is choose the Fc. So what Fc do you want? Mouse, rabbit, human, and order. Where do you order? It's on the same page. It's here at the bottom of the page. Here is the information for ordering. If you click on the mouse link, as is explained above, you will get to order this antibody with a mouse Fc. You can pay directly with a credit card, and your order will be processed automatically. The cost for this is 200 Swiss francs. We know that we all need cheap antibodies in academia because we're poor, and 200 Swiss francs, I think, which is more or less 200 euros or 200 US dollars, is an affordable price, even for academia. You get this antibody if it's the first time that it's produced and has never been used. We recommend that you test it. I mean, yes, there is a paper that says that this antibody works. You may want to test it in your own conditions, and then you can use it. And then we suggest very strongly that you publish the result of this test. And again, this is what Antibody Reports is for. So once you've done this test, rather than just keeping it hidden somewhere in the lab book, publish it. This would be a very good project for your newcomer graduate student. And she will publish his first paper that way. Who knows, this may end up being her most cited paper ever. Now, what happens if there are no antibodies against your favorite protein? Okay, nobody has developed an antibody against your protein. It's too new, too marvelous for that. What are your options to get recombinant antibodies anyway? Option number one, there is actually a sequence that you know about of an antibody that's supposed to recognize this antibody. It's been published, it hasn't been yet added to ABCD. You can submit the sequence and then you're back to square one. You can order the antibody, just like for any other ABCD antibody. Possibility number two, there is no known sequence, but there is a hybridoma that produces this antibody. You can sequence the hybridoma, or you can ask us to sequence the hybridoma. The cost is 500 francs. It's not going to break your budget. And then you will get the sequence in ABCD. You can order the antibody. And why would you care ordering an antibody as a recombinant antibody rather than as a hybridoma supernatant? Because then you will know the sequence, so you know exactly what you're working with. And because also, now you can choose. If you want to have it rabbit antibody, if you want to have it human antibody, you choose the Fc. But then there are many antigens for which there are no recombinant antibodies and no hybridoma. So what you can do is use our discovery facility. Your first aim is to choose the antigen, and you must choose the Fc that you want to work with. And then we will do the screening for you. I will describe the method rapidly. And the cost will be 2,000 francs. So how does it work? We start from a library of phages, either human antibodies or llama nanobodies. You may know that there are two different formats of antibodies. Those that have two immunoglobulin domains, typically human or mouse antibodies, or those that have only one immunoglobulin domain, like llama antibodies, also known as nanobodies. We have both libraries, we can screen both. And then we can select for you antigen binding phages. Beware, the selection is done in vitro. It means that you control the selection conditions, so you know exactly what your antigen is and in what conditions it was bound by the antibody. You can deplete some antibodies before selection. So let's say you want antibodies that recognize a phosphorylated peptide, but not the non-phosphorylated peptide. You first deplete all the antibodies that recognize the peptide, and then select those that recognize the phosphorylated peptide. But it also means that you must choose carefully the right antigen for you. If your aim is to work with an antibody that recognizes a full protein, well, then probably you should use the full protein as an antigen rather than a peptide. If, on the contrary, you want to work with peptides, modified peptides, select the right antigen. We will select antibodies that bind your antigen. We will sequence the corresponding antibodies, at least 10. We will produce and test these antibodies by ELISA, and then it's back to you. You will get 5 mL of each antibody back to you. And then again, don't forget, test them, publish them. This is a service you're doing for the whole community also before you start using them. You've developed a new antibody. This antibody is available for you for the rest of your life, for everybody else also, which means that you really have made a work that benefits the whole community. I'm already coming to the end of this presentation, and there are a few points that I want to stress before I end. First, recombinant antibodies will replace classical antibodies. The improvement of science reproducibility commands that we do this. The sooner, the better. But even if you don't want to participate, you will be forced eventually. In 10 years, 20 years from now, people will laugh at the idea that people used antibodies without knowing what they were, or an undefined mixture of antibodies produced in a rabbit. Second point, the ABCD project, as I described to you, is a community project. It's academia to academia, it's not for profit, it's open access. It's really a project that was developed for the community. And basically, my third point is we need you. The community project needs the involvement of the community. You need us. There is a lot of expertise for which you need help. Contact us. There are no limits to how ready we are to collaborate with any group in the world in academia. We're ready to pass our libraries of pages. We're ready to pass all the material we have to anybody. This is a project which is only meant for developing recombinant antibodies in academia. Contact us. Thank you.
Thanks, Pierre, for your informative presentation. As a reminder, we're going to hold questions until the end, but please send them in at any time using the GoToWebinar questions panel, and we'll ask them after our next presentation. Speaking next will be Dr. Michael Fiebig. Dr. Fiebig is the Vice President of Product Portfolio and Innovation at Absolute Antibody, which specializes in antibody sequencing, engineering, and recombinant expression. With a background in both wet lab and bioinformatic methods, Michael developed the first version of Absolute Antibody's high-throughput hybridoma sequencing platform, forming the basis for a realistic approach towards converting every hybridoma-derived antibody into a recombinant antibody. I'll now turn the floor over to Michael.
Yeah, thank you very much for the kind introduction, and thank you very much to Pierre also for a great talk, which I think really sets the scene for what I'm going to be talking about. I would like to talk to you today about sequencing existing antibodies. So quite deliberately, we're not going to talk about sequencing in the context of antibody discovery, sequencing phages, or single B-cell sequencing. I'm going to talk about sequencing the antibodies you already have, specifically focusing on hybridoma sequencing, but then also looking at antibody protein sequencing and going beyond. But first, let me give you a little bit of background about Absolute Antibody. We're an antibody supply and CRO founded in 2012, and we've rapidly grown to about 60 staff across 3 locations. And we were founded with the vision to make recombinant engineered antibodies and related services more accessible to the wider community, particularly those in research and diagnostics. People working in the biopharmaceutical space have generally already sequenced and made their hybridomas recombinantly, but those at the lab bench at universities and people making diagnostics generally seem to be lagging behind a little bit. And in order to achieve this, we've industrialized 3 core technologies which we apply to our services and our catalog. And these are hybridoma sequencing, antibody engineering, and transient expression of antibodies. And I would like to share some of our experiences that we've made whilst developing a hybridoma sequencing platform with you in the next few minutes. Now, what is a hybridoma and why are we falling out of love with it? Many of you will be aware that it's a fusion of an antibody-secreting B cell from an immunized animal with a myeloma cell. And from that, you get an immortalized antibody-producing cell that you can culture in vitro. And, you know, in an ideal scenario, that's actually quite good. However, there are a range of problems with hybridomas that stem from how they're created. The genetic makeup is a fusion of a B cell with a myeloma cell. A myeloma cell is a cancer cell. So you have an inherent genetic instability, which leads to genetic drift or other changes that can happen to the antibody that the hybridoma is making. Also, there's a transcript contribution from both the myeloma cell and the B cell, and myelomas can actually bring with them antibody transcripts themselves. So you have a mixture of transcripts in there. There can be multiple fusion events. These were reported right in some of the very first papers about hybridomas, that you haven't just fused 2 cells, but 3 cells or even 4 cells, again, creating a more complex product. Now, from a practical point of view, hybridomas can sometimes be difficult to adapt to serum pre-growth, so you're stuck using lots and lots of animal-derived materials like serum. And also sometimes hybridomas don't grow in vitro, so you have to culture them, grow them up by ascites, where you take the cell line, inject them back into the peritoneal cavity of an animal, and then you harvest the juices that accumulate around that growth. It's a pretty gruesome process. And clones can die off or be lost. So maybe they were frozen down poorly, maybe there was an accident in liquid nitrogen, or someone just retired and their entire career's worth of hybridomas were disposed of. All these things and lots more reasons are why you should be sequencing hybridomas. Now, we've been sequencing hybridomas since the 1980s, and there are 2 main approaches that have really been the workhorse of this industry. One is V-region PCR, and the other one's 5' race PCR. What you have here is on the left-hand side of the flow diagram, you use degenerate primers that amplify up the variable regions of the antibody. These are the antigen binding domains. Or on the left-hand side of that flow diagram, you've added a homopolymeric tail to the transcripts, and then you use that from one side to prime a PCR reaction, and then primers annealing to the constant domain. Whichever one your tool of choice is, once you've amplified up the variable domains, these are generally cloned into bacteria expression, into bacterial vectors, grown up, the bacteria are streaked out on a plate, colonies are picked, grown up, and then DNA extracted, and they're then finally submitted for sequencing by methods like Sanger sequencing. Now, this is the method, these are the methods that the majority of people sequencing hybridomas use. And there are many advantages to this. It's got very low consumables costs, and it's relatively simple. There are quite a few steps in here, but fundamentally these are basic molecular biology steps, and it's something that you can set up. However, there are some disadvantages to this. It's fairly low throughput and does require a little bit of tinkering to get the conditions right. And it's not something you can easily automate if you want to sequence every hybridoma in the world. It's also error prone. These primers can introduce small mutations. And also in early rounds of the PCR reactions, there are mistakes these can propagate through. And finally, it's quite difficult to reliably sequence non-monoclonal hybridomas with that. What I mean with those non-monoclonals. We were recently involved, a couple of years ago, involved in a study where quite a few contributors looked at over 180 hybridomas. And when we analyzed the composition of those, it became apparent that only about 2/3 of these hybridomas actually only had one heavy and light chain pair. The others had additional light chains, additional heavy chains, additional light and heavy chains. And what this results in, if you just have one additional light chain, you actually get 3 products coming out of your hybridoma. if you've got two additional products, then you see you get quite a collection of different products, and these are going to affect the performance of an antibody. For example, here we switched some of these complex ones to just the active ingredient combination of heavy and light chain, and you can see the results you get with the recombinant version. It's just a little crisper and a little bit better. And you can read up all about this in this paper here by Bradbury et al. From a couple of years ago. Now, this affects the sequencing because there is no correlation between abundance of a transcript and functionality. The right transcript that you might be looking for might be there at 1 in 100 or 1 in 1,000 ratio to some aberrant transcript that is flooding your reactions. So how many colonies do you want to be picking? And it's also not always clear whether something is a non-functional transcript or not. These methods focus on just the variable domain regions, which emits some valuable information that can be contained in the constant domains. And now you might be asking, well, does it really matter? Does this really cause problems? It does. If we're looking at public sequence databases, you can find many artifacts. These can be in patterns, these can be in papers, these can be in crystal structures, where things like well-known myeloma-derived sequences have been threaded through densities that don't correspond at all, but we're still interpreting meaning into finding these sequences in there. And so people have just identified the wrong sequences. Also, lots of non-functional sequences and non-expressing sequences, which we've come across trying to make some of those at Absolute Antibody as well. So really, we needed a better approach to not only be able to sequence our own hybridomas, but also those of our clients. And what we decided was to go along a next generation method, or rather a whole transcriptome shotgun sequencing method. And what we do here is we take out the total RNA from a hybridoma, we fragment it, and we sequence all those little bits of cDNA fragments, and then we assemble those together, and from that we identify the antibody coding transcripts from the entirety of the transcriptome of the hybridoma. With this, you don't need specific primers. This all just runs of random primers. It's compatible with any species and isotype. It's high throughput, fully automated data analysis. And even when a transcript is at very, very low abundance, you can still detect it, assemble it, and at least start to consider it. You can rescue dead hybridomas, because if the RNA is a little bit chewed up, well, we're going to fragment the RNA anyway. So that is actually possible. And you actually get information on the full antibody sequence. Well, very large parts of it. There are other approaches trying to combine this sort of NGS sequencing approach with race PCR. So first amplifying up the variable domains and then sequencing this. This is something we found to have somewhat lower sensitivity. Again, you need information about, you need to focus your protocol on particular species and isotypes. And sometimes you don't know. You'd be surprised what kind of hybridomas can show up in front of you. And again, this would only identify variable domain sequences. So we believe going in a total transcriptome is a little bit superior. Now, we believe that with this next generation hybridoma sequencing platform, we've developed a technology that is reliable enough and of sufficient throughput to sequence every hybridoma out there. Let's look at some numbers. This graph shows the cumulative number of hybridomas we've sequenced, and I probably didn't need to put in the line to show where we switched from old school sequencing to next generation sequencing. I think the increase in throughput is quite striking as is. And also looking at numbers we did per year, this is only going up to 2019, but we are now doing well over 1,000 clones a year. And yeah, before we switched to NGS sequencing, our team sequenced about 300 clones in 4 years, and that was a lot of work. And again, this same team is now, as I said, doing well over 1,000 clones. And quite honestly, the bottleneck is not processing the samples, is actually getting hold of enough clones. Because we believe we could definitely be doing over 10,000 clones a year, and that's just with the same team that was doing 300 clones before. And all the while through, we're still harnessing the advantages of being able to sequence any species, highest possible quality, and getting full sequences. Now, there are some disadvantages though, and this process is a little bit slower for small sample numbers. If you have 1 or 2 hybridomas and they behave well as you're processing them, you might be able to sequence them in a week or in 2 weeks. But if you're looking at sequencing entire collections, or they're a little bit temperamental, then this method that I was just showing should be considered. And then you've got somewhat higher operational costs. However, if you look and compare pricing amongst commercial suppliers of hybridoma sequencing services, I don't think you'll necessarily pick out who's using old school methodology and who's using a more modern methodology based on pricing. Now, what about ascites? I touched on the fact that there are clones that can only be grown in ascites, and that's not something I believe people should be doing anymore. Now, sequencing these clones is absolutely no different to sequencing a clone that will grow in vitro. We're just cracking out the RNA and we're sequencing it. I've got an example here where we sequence a hybridoma that would be grown in ascites, and actually when we sequenced it, we found no secondary antibody transcripts, so it was completely clean. But when we made it recombinantly and looked at the performance in ELISA, we actually found just making it recombinantly actually worked better. So we've killed 2 birds with 1 stone here. We've got a better product, and we don't need to do ascites anymore. So, no more excuses for hybridomas. Sequencing approaches are reliable enough and work for any hybridoma. And we've really reached the throughput required to sequence every available hybridoma. But what about lost clones? Now I've mentioned hybridomas that are non-viable anymore, but might still have some material left of the hybridoma. But there are cases where people have actually just not got access to the hybridoma anymore, and all that's left is a little bit of protein. Don't despair. It is possible to sequence antibody protein. The technology's really come along a lot in the last years. So what you do here is you take the antibody protein, fragment it by enzymatic digest, different types of enzymes to get different types of peptides, and these are then sequenced by mass spectrometry. And really here the innovation is that the de novo peptide sequencing, the peptide identification algorithms have really evolved massively in the last 5 years. And from these peptides, you can start assembling whole antibody sequences. And you only really need about 100 micrograms of pure protein to sequence an antibody. And I do know of some people who've managed with about 20 micrograms, but that was very clean antibody. So before needing to go and raise a new antibody, whether it's by an in vitro method or in a hybridoma, maybe you've still got some of that antibody and you can just recreate it and thereby be able to continue the research legacy of that clone. And a final, what about valuable polyclonal antibodies? I think we all know there are some fantastic polyclonal antibodies out there with performance characteristics that you cannot recapitulate by a monoclonal. Now, current sequencing approaches that try to sequence polyclonals are looking to combine transcriptomic and proteomic approaches. So we're sequencing some B cells from the animal we're immunizing and taking serum to bring these data sets together and identify some lead candidates. Now for that, you do really need to have access to the animal, and maybe it's a little bit of an arduous task to do this. The holy grail really is here, could you just take a polyclonal that you know worked very well, and could you just reconstruct that by mass spectrometry? And I think this is in many respects the one of the missing keys in this journey of being able to take every reagent and make it recombinant. And the technological developments in this field are also progressing very nicely. And I think maybe already in 2021, we're going to see some very exciting developments in this area. So in summary, antibody sequencing is a good thing. I think whether you're worried about quality, reproducibility, engineering options, animal welfare, all of these or many other reasons, I think we can all agree to that. Now, classic hybridoma sequencing is limited in terms of quality and throughput. But this next generational shotgun sequencing approach not only brings you the quality, but also the throughput to allow us all to sequence every hybridoma that is out there and bring these to the next age of antibodies, if you want. And we can do so reliably. Now, protein sequencing approaches are available for monoclonals at the moment, perhaps as a last resort. But again, I think the polyclonal antibody protein sequencing is one of those little missing blocks in the journey here, and I'm quite confident we'll have something available for in the industry for this. Right. And that's all I wanted to say. Thanks again to the organizers, and thanks also to my colleagues who do all the sequencing. If you have any questions about the things I've just talked about, or you want to see how this could be applied to your projects or problems you might have with your antibodies, you can contact us at support@Absolute Antibody. And I look forward to answering any questions you might have.
Excellent. Thank you so much, Michael and Pierre, for some really wonderful presentations to tie things together very nicely for the webinar series that we've enjoyed over the past few months. We do have a few questions in the question box, and again, so I'll remind folks to please utilize the question function on WebEx to enter in questions. The first one, I suppose, is for Pierre, but probably for either. There's, reference to the ECFAM peer review and report associated with some concerns over that review, and also the questions around concerns regarding synthetic antibodies for their use in therapies, as well as challenges regarding scale up, perhaps associated with cost and time. Do you have any comments or thoughts on either of those, and how we might persuade researchers to implement these types of approaches in their laboratories?
Maybe I can start. So I'm not sure if I absolutely understood your question. So, but as I understand it, it's a question about the scalability of antibody production.
Correct.
And the quality of the reagents obtained, and how to make it a standard type of reagents. On the scalability, I think it's true. It's important to realize that it's different to produce huge amounts of very pure antibodies or small amounts of relatively pure antibodies. On our side, we have worked a lot on scaling, but we were scaling down. We wanted to produce very small amounts of antibodies, 5 mL maximum, so that we could send the whole preparation to the laboratory that's ordering it, and the cost should be limited. And we didn't want to go through, of course, generating stably transfected cells. But of course, the concerns are very different if you want to scale up and if you want to get more pure antibodies, if you want to use them, especially if you want to go for therapeutic use or even just diagnosis use. And then it's a completely different story. And I think at least we would not be competent for that part. Michael?
Yeah, I think it touches on a very so something. How do you approach making lots and lots of recombinant antibodies? And especially how do you approach making lots of different versions of the same antibody with different FC portions, etc. We've actually found that transient expression for the reagents market is definitely the way forward if you're making these. There's no point making hundreds and hundreds of stable cell lines. One reason for this is that transient transfection methods have actually gotten really, really good. And now there are commercially available cell systems out there that can get you up to grams per liter. We use our own platform, which can hold its own. I don't think it's as good as some of the very best ones out there, but we can make grams, tens of grams of antibodies for research within a matter of weeks. Do we do that all the time? No. Generally for the research reagents market, you might need a few milligrams in stock, maybe tens, maybe a few 100 milligrams in stock. And already from that, you can cater to people requiring smallish quantities, 200 micrograms for their flow cytometry experiment, for their Western blots, but also to people who might need 50 milligrams or 200 milligrams for an in vivo study. And in our experience, we've standardized all the purification processes as well. And so if you have that setup, you can actually already get very, very, very pure material that can hold its own. You know, we're talking 99% pure, 99% monomer fraction, etc. So there are, of course, additional costs of cleaning things up more and more. But I don't think it's a challenge that impacts the profit margins unreasonably. And you know, we're running this as a business, and it's possible to do this.
Excellent. Thank you both. Very helpful answers. I suppose this is a question for Michael. How do you handle possible intellectual property issues, both in terms of submission of the antibody for sequencing and ownership of that sequence?
Yeah, so it's a very good point. So there are two scenarios. Either it's an antibody that we've licensed for our catalog, and in that case, the antibody and the sequences remain property of the people who license the antibody to us. We do not disclose the sequences of antibodies to third parties ourselves. The other scenario is if someone comes to us and wants to just have their antibody sequenced and maybe expressed, maybe not. There, we just provide a fee for service. The antibody remains the property of the client, and they're free to do with it what they want. We don't take royalties at the end, and these antibodies do not end up on our catalog either, unless you decide you want to license them to us as well.
Great, thank you, Michael.
Yep.
Pierre, your talk, you mentioned your services primarily for academic researchers, but do you have advice for a company that might be looking to become involved in developing and supplying antibodies using these in vitro methods? And I guess a similar question would be, could the pharmaceutical industry access ABCD services? So
the short question is yes. We're willing to collaborate with anybody interested, and we have been working for now quite some time on this pipeline of discovery. So we have our own libraries of antibodies. We have all the expertise for selection. We have, I mean, not so many people have, like we have done, selected antibodies against hundreds of different targets of all different kinds. So yes, we do have a lot of expertise, and we would be just absolutely willing to share it with companies.
Excellent, thank you, Pierre. And I guess a follow-on question for you, what would be the expected time frame for production of a recombinant antibody after submission of the antigen of interest?
As you may know, the discovery process itself is actually pretty fast with phage display. The discovery process takes about 2 weeks. So from the time you start your selections, so you have your antigen ready, and you can work with it, takes you 2 weeks to get the antibodies, let's say another 2 weeks to test them. So within 4 weeks, we can get antibodies. Now, of course, this is depending on how full the pipeline is at the moment. So that may vary in time. The minimal time, I would say, is 2 weeks.
Wow, okay.
4 weeks, yeah, it is. 4 weeks from
Okay.
the time we get an antigen to the time where we have specific antibodies against it. I think it's really important for people to realize that when you develop a new antibody, the characterization takes a significant amount of time. So sometimes people very naively think, I'm going to develop this, get this antibody and it's ready to use. No, you have to characterize it. You have to make sure that it actually recognizes your protein in the conditions where you want to use it. If this is an antipeptide that you have generated, it may recognize the full protein or not. And that's really important to spend the necessary time to characterize these antibodies if they're created for the first time. So that part is probably the part that takes the longest, not the generation of the antibodies. And that has to be done by the lab that's the final user of the antibodies, otherwise it's pointless. So we do in ELISA in vitro characterization, but then eventually if you want to use it on cryo sections, you have to make sure it works in cryo sections.
Excellent, thank you, Pierre. That's quite impressive. Michael, a basic question perhaps, but could you revisit your, you referred to your next generation sequencing approach to be able to rescue dead hybridomas. Could you expand on what you meant by that and exactly how that works?
Yeah, so many people have hybridomas that they dig out of the liquid nitrogen and they try and grow them up again in the cells. They just float around and they don't do anything, and fundamentally just a lot of cell debris there. And for us, so when we receive such a sample, we can still extract mRNA, or at least mRNA fragments, from these cells. Most of the time, the nucleotides in there are still somewhat intact. And so because we're not reliant on actually having lots of intact RNA, but you can just have bits and pieces, it doesn't matter to us in our sequencing approach because we're still going to be piecing all of them together anyway. Now, for hybridomas that are dead, we've got or at least non-viable, we've got about a 50% success rate at the moment. So it doesn't work all the time. That's why something like an antibody protein sequencing service is a good backup. But before we throw our hands in the air and give up, it's always worth giving it another shot. If you're using traditional hybridoma sequencing methods where you're actually trying to do PCR, if the template is chewed up or fragmented, you're not going to actually get priming of the PCR reaction. And so there you struggle a little bit more. But with this next gen method, I think we overcome that a little bit because we're not reliant on necessarily intact RNA.
Excellent, right, thank you, Michael. Pierre, I suppose this question came in as you were describing your turnaround time process. For well-known antibodies, does the characterization validation still take place?
Say again, does the characterization?
Does the characterization or validation of the target still take place in your process?
I'm not sure if I understand what you mean by characterization of the target. I mean, maybe this touches on actually a really important issue, which is the choice of the antigen which is being used. You're a laboratory, you want to use an antibody for histology detection, you select it against a protein of your choice. So that's, maybe you actually will want the protein to be fixed to reproduce the conditions in your histology section. And all these things, the final characterization of yes or no, does the antibody work in your final laboratory conditions, that's the work of the laboratory. But what we will do is we will get the antibody. So if it's a simple one, we will synthesize it ourselves, like peptides. But if it's a complicated one, we will receive it from the researchers. And then we will handle this. And this is the antigen, whatever it is. It's fixed, it's not fixed. It has been modified in a way or another. And this is what we will work with, and we will make sure that the antibodies, of course, bind this antigen in vitro. But then again, is that same antibody going to work in a histology section? And maybe there will be some characteristics. Does it work in immunofluorescence? Maybe only in methanol fixed cells and not in paraformaldehyde fixed cells, for example. And that type of expertise can only be done by the final user, in our opinion.
Excellent. Thank you, Pierre. It looks like we have gone through all the questions received so far. Did you have a question, Pierre?
So maybe I can ask a question to Michael. So Michael, you mentioned sequencing, protein sequencing of polyclonals, which, as you say, is a fascinating development. But how do you handle the fact that you have all these heavy chains and all these light chains, and which heavy chain is going to be matched with which light chain?
Yeah, that is the big question. So within a polyclonal, well, polyclones can be very, very, very diverse. We can appreciate that. The idea is that there are, I guess, families of clones within this. So you might have antibodies that differ in a couple of positions, but fundamentally they've all derived from the same precursor. And so if you can pick out members of maybe 3-4 different families and start off with that, the next step is then which ones might be the most abundant, perhaps you can, of each family, maybe you can focus in on those, and then looking at pairing. Now on the pairing, I think there are some clever people trying to figure out whether there might be predictive aspects about this. With some of the collaborators we've worked on this, we've actually just said, you know what, let's do a grid expression. Let's take 10 transcripts here and 10 transcripts here, and let's pick out which ones, first of all, express. That's always good when all the bad pairings don't express. That's actually brilliant. And then of the ones that expressed, which ones are actually functional. And you know, some of these early attempts we did, I think we did 10 by 10 expression, and we got 3 active ones, 2 of which actually shared a light chain, but were both fantastic binders. So you know, these were baby steps. But I think as this evolves further, if all you need to do in the end is do a 10 by 10 expression and you get 5 good binders, I think that'd still be pretty amazing. So yeah, a bit of brute force sometimes.
Thank you, Michael.
Yeah. And can I ask you a question back?
Is that
yeah. Absolutely.
So you work with the mini body format where you convert things into scFvs and then put them on the Fc domain. 2 questions about this. One, do you ever find that maybe like not all mouse antibodies really like to convert into single chain Fvs, or and in turn, you know, yields can be particularly low with those? And the other thing is, do you have a lot of issues with aggregates where different scFvs exchange light chains and then sort of you have 2 IGs chained together?
So let's answer the first question. First question, as a rule, we get yields. We haven't optimized so much in terms of yields because our idea was that most users need 200 micrograms of antibody for their regular laboratory work, Western blots, immunofluorescence, histology, whatever. Usually they have enough to store it for years with just one delivery. But yeah, a percentage of antibodies have problems in producing in the scFv format. I would say 10% of them maybe, but I have 10%, maybe 20, but not more. And I would not say that, but I haven't checked actually, I could check. I haven't noticed that mouse antibodies were particularly difficult to handle in that frame. Nanobodies, single chain, heavy domain, those are really give super heavy, super high yield, but no major problems. You had a second question, which was?
Was whether you encounter issues with light chain swapping between different scFvs. So you might have a monomer sort of heavy chains with single chain of Phis, and then higher order aggregates of those.
You would detect these in non-reducing gels, or
gels can be probably, but better would be like a size exclusion step on this. So we sometimes see that multi-specific.
Practically, we didn't encounter a problem there. So I would guess that whatever works. With the antibodies work in the end, but whether there is a fraction of higher order aggregates that either are eliminated or lost at some stage, or left but somehow less active, we have not noticed that as a major issue.
Cool.
Maybe we haven't looked hard enough. Thank you, Michael. Hey, thank you to all the people who attended this webinar. It's a real pleasure to be able to present this project to all of you.
Yeah,
excellent. Thank
everyone.
Yeah, thank you both. Really fantastic presentations, and I don't mind saying the last 10 minutes have really been sort of one of the goals of this entire effort, is to bring some of these experts together for future collaborations and conversations. So this is really wonderful. With that, this concludes our fourth of 4 webinars that we had on the calendar for this year, 2020. We possibly will convene additional webinar series on this topic in the new year. I would certainly encourage folks that have participated, have been involved, if you have thoughts or ideas around the series, to please bring those forward. You see on the screen there contact information for Catherine Groff and myself as well, so please feel free to reach out. As Catherine mentioned at the outset, all of the slides from today's webinar and the prior webinars are archived on the PETA International Science Consortium website. You can see the hyperlink there. With that, that brings us to a close. I do want to take just a moment to really thank Sasha Yusufovic. Excuse me, I'm sure I destroyed your name, Sasha, but thank you so much. She's behind the scenes making sure everything runs smoothly during the series. It's been wonderful. And absolutely thanks to Catherine Groff for just a fantastic job of doing a lot of heavy lifting and pulling these experts together for this series. It's really been a wonderful series, and we're excited to see things that develop. With that, we'll sign off, and thanks everyone for participating.
Thank you, Dave.
Thank you very much. Have a nice day.